I'm Stephen. I have a background in cognitive neuroscience research and data science.
Hi, I'm Siddharth, a comp bio and CS student at CMU.
Neuroscience runs on MRI data, and MRI data is a mess. Proprietary tooling, scanner fingerprints, hundreds of pre-processing steps, and after hours of work, you find your model is tracking which scanner took the scan and not the patient. It gets worse downstream. In biomarker discovery, the labs collecting scans and the labs finding biomarkers don't even share tooling. The wet lab inherits data it can't rederive or fix, and every handoff stalls. You never get one clean pipeline. So we built NeuroAD, a Claude Science plugin that closes this gap. It ranks candidate biomarkers using NeuroJEPA, then runs our silent failure guard on the scans to catch bad data before it costs you. One loop from hypothesis to a verified wet lab candidate, all coordinated through Claude Science.
I'm a neuroscience researcher, and I go into Claude Science and connect our tool NeuroAD. I have an Alzheimer's disease hypothesis I would like to test. I want to validate which candidate proteins are worth investigating further. I see which experiments are not worth pursuing based on confounders. And after the decision tree analysis is complete, I land on the top candidate. The top ranked candidate has the AUROC with the different confidence intervals, as well as other proteins that have been ranked below it.
Once candidates are identified, we automatically run a panel of MRI quality control tests to catch scanner artifacts and pre-processing issues before they cause silent failures. Here, we're checking alignment against expectations using classical MRI tools. Given the number of possible cofounders, this scan should be culled. I'll flag it by double-clicking anywhere to drop a pin, then add a note to ask Claude about it. The surrounding region is sent as context automatically. I save this response to the notes drawer, and these notes pass straight back to Claude Science as context for downstream tasks.
We see relevant drugs for further evaluation and experimentation, as well as why our top candidate was chosen, provenance and reasoning, as well as offshoots and confounders that did not survive our gauntlet. Let's drill down into the final protein details. This is what the protein structure of our top candidate looks like. This is taken from the AlphaFold database, and we are left with a testable wet lab experiment completing the translation between the human fMRI world and the wet lab world.